Welcome to Pihu classes. Today we will learn osmosis.
Osmosis is how water molecules move across a semi-permeable membrane that separates two solutions. It can be thought of as a passive diffusion of water, and it requires no energy. When water molecules move like this, they end up equalizing the concentrations of the solutions on either side of the membrane. This is possible because a semi-permeable membrane, like the cell membrane for example, is kind of like a sieve with pores that let small molecules like water across, but not larger molecules or ions like sodium and chloride. So let's say that we're looking at a lab beaker that's filled with a saltwater solution, and we separate it into two compartments, A and B, with a semi-permeable membrane in the middle. Now, first off, the salt, which is sodium chloride, will separate out into sodium ions and chloride ions once it's in the water. And since the concentration of sodium and chloride ions is the same on either side of the membrane, we say that A and B are isotonic to each other. Now, inside the two compartments, water molecules and sodium and chloride ions are moving around and bouncing off each other. It's kind of like two big dance parties happening in two adjacent warehouses that are connected by doors that are semi-permeable, meaning that the water molecules can get through but not larger sodium and chloride ions. Now, some of the water molecules might go through one of those doors, to go from party A to party B. And some water molecules might go in the other direction, from party B to party A. But the truth is that water molecules aren't particularly drawn to either compartment, because crossing the membrane to go one way is just as easy as crossing the membrane to go the other way. We call this point equilibrium, and in this particular state, the net movement of water across the membrane is zero. But what if we were to add some additional salt, or sodium chloride, inside only compartment A? A now has more solute than B, and we would say that A is hypertonic compared to B, and conversely that B is hypotonic compared to A. And what we'd notice over time is that because A has more sodium and chloride ions, its osmotic pressure increases. The result is that we'd see a bigger net migration of water molecules over to side A. We say net migration because water molecules are still going back and forth between the two sides, but overall more water molecules will end up staying on side A. Once there's enough water that the concentration of salt on the two sides is equal once more, then the net movement of water across the membrane goes back to being zero, and the two sides are isotonic to one another again. So far, so good. But now there's the bigger question. Why do water molecules end up staying more on side A? The answer to this has to do with kinetic energy and entropy. Kinetic refers to the fact that water molecules, as well as sodium and chloride ions, have a tendency to want to move around. And entropy plays a role because this movement is disordered, or random, so that over time the water molecules and ions move every which way. You can think of each molecule or ion like a toddler. You know it's going to move, but you can't really predict its next move, or where it might end up 2 minutes later. Now, going back to the experiment, when we initially added more salt, there were relatively more sodium and chloride ions on the hypertonic side, or side A. Now we know that sodium and chloride ions are too large to pass through the pores in the membrane. But in addition to that, and this is the important part, the ions actually end up slightly blocking or interfering with water molecules that want to go from one side to the other. That's because the ions are so large that as they're bouncing around, they literally get in the way of the door. It's like a party goer that's unable to leave a party but also blocks the 5-foot area around the door, making it hard for others to leave the party as well. But remember that these ions are not physically stuck to the hole between the two sides. They're just knocking water molecules in that area out of the way through their random bouncing movements. To make matters worse, the sodium ions are positively charged, and they actually attract the slightly negative charge of the oxygen atom in the water molecule. Similarly, chloride ions are negatively charged, and they attract the slightly positive charge of the hydrogen atoms in the water molecule. If these water molecules are somewhat attached to an ion and can't get through the membrane, they'll also be hindered from getting through the membrane. So in short, the ions that can't get through interfere with the water molecules from leaving the party. And since the concentration of ions is higher on side A than side B, more of this interference is happening on side A. As a result, more water molecules are ultimately able to get from side B to side A than in the other direction. All in all, this means that when there's a difference in concentrations, water molecules move from an area of low solute concentration to an area of high solute concentration. Put differently, they move from the hypotonic side, where there's relatively more water, to the hypertonic side, where there's relatively less water. Now, it just so happens that osmosis is super important for our cells. For example, let's say that we put a red blood cell in a hypertonic solution like super salty water. Right away, there would be a net movement of water molecules out of the cell, leaving our cell dried out and shriveled. On the other hand, let's say that we put a red blood cell in a hypotonic solution, like pure water. This time there'd be a net movement of water molecules into the cell, making it swell up and eventually burst, neither of which are good ways to go. Just like Goldilocks, our cell would prefer something in the middle, which is an isotonic solution, like the intravenous fluids that are used in hospitals. These fluids have sodium chloride in a concentration that's similar to the solute concentration inside the cell. In this situation, the net movement of water inside and outside of the cell is zero, so the cell continues to live comfortably. Alright, as a quick recap. Osmosis refers to the movement of water molecules across a semi-permeable membrane that separates two compartments of fluid. It can be thought of as passive diffusion of water, and it requires no energy. The side that has more solutes will prevent water molecules from leaving, and as a result, there'll be a net movement of water to the hypertonic side. Over time, the movement of water molecules from the hypotonic to the hypertonic side will equalize their concentrations.
Thank you.